Hey everyone, welcome to chapter 16. This is whenever your immune system goes wrong. We're going to talk about the 4 categories of hypersensitivity, or the 4 categories of overreaction to antigens, how allergies develop, how they can be counteracted, transfusing compatible blood, what happens if you do incompatible blood. Describe the mechanism of type 4 hypersensitivities and why they are delayed, and you will understand it in a moment. The theories on how autoimmunities develop in the first place, and we're going to talk about the difference between primary and secondary hyposensitivities. So, hypersensitivities and hyposensitivities. Okay, so the top is talking about the hyposensitivities. That's where you're not reacting enough, right? So hyposensitives, you're not reacting enough. So here we have a primary immunodeficiency. So that's somebody who had this to begin with. So this is from the get-go. It's usually genetic. This person has severe combined immunodeficiency. It's a genetic disorder. It's called SCID. It's another boy in the bubble. Then we have secondary immunodeficiency, which is where it acquired somehow. And then we have, like, include someone with AIDS. Then we have the hypersensitivities, and we're going to go through each of these, and we're going to start with these actually. But yeah, type 1 is like allergies and anaphylaxis. Type 2 is going to be antibody mediated, and it's going to have a lot to do with blood incompatibility. Type 3 is going to be immune complex development, and then type 4 is going to be cell mediated. So antigens that elicit hypersensitivity are going to be either exogenous or endogenous. These are terms that you should be familiar with by now. Exogenous from the outside, endogenous from within us. So with stuff that comes from within us that we're, hypersensitive to, that would be an autoimmunity. So these are some examples down below here for each of these setups. If you want to look through these, we'll be going through most of these and giving examples for each one in the following slides. So type 1, sorry, my butt is sore. I've been doing these videos, man. Type 1 allergic reactions. We have atopy and anaphylaxis. So I do want you guys to know the difference between those two. An allergy is anything where you have an exaggerated immune response that is manifested by inflammation to usually something that you shouldn't be having an immune response to, at least not that strongly. Allergens include antigens that do not noticeably affect non-allergic individuals. So that's another way you can compare it. Okay, so like the majority of the population doesn't react to it, then and you are, then that we call it an allergen. Right. Similar physiological, I don't know what, similar to what? Allergy, okay, whatever. And it has immediate onset. Similar, what? Similar. I guess if we're talking about atopy versus anaphylaxis, I guess they do. Okay, I guess that's what it's saying. Atopy versus anaphylaxis, which we haven't introduced those terms yet, and we're about to, but they have similar immediate onset, and they're associated with exposure to a specific antigen. I guess that's what I'm trying to say, but I don't know always what I wrote last semester. So we have 2 levels of severity of allergies. We have atopy, which is local, and anaphylaxis, which is systemic. An example of a local allergy, hay fever, cat, being allergic to cat dander or whatever, dogs, all of that. And then anaphylaxis is systemic, and I mean systemic. So that can be fatal. It can obstruct your airway and cause circulatory system collapse. How do allergens get into our bodies? We have these 4 kinds, and I do want you guys to be aware of what the 4 kinds are and be able to at least give an example of each of the 4 kinds. We have inhalants that you breathe in, obviously. Ingestants that you ingest by typically eating or drinking. Injectants. That does not just include things that are coming in through a needle. We're also talking things that are, like venom from wasps and bees and stuff. Okay, anything that's been put in under your skin forcibly. And then contactants, anything that actually like just it touches your skin and it causes you to have allergy. So some common ones, inhalants like being allergic to pollen, ingestants like being allergic to peanuts, injectants like being allergic to bee venom, and contactants like being allergic to latex. Okay, pretty common stuff that we've been kind of dealing with our whole lives. We just don't talk about them in these terms. Okay, so how do we get to this point? What's happening here? We have a T helper cell. Remember, they activate our other cells, right? And we have this allergen that's come along and it started to right. And it started to interact with the T cell, B cell. The T cell is going to, helper cell is going to activate the B cell and trigger it to respond to whatever this thing is, this allergen is. Then it's going to produce antibodies against that allergen. This is typically going to be IgE when this type of reaction, an allergic reaction, is going on. Allergies, you're going to produce IgE. It's a type of antibody. It's like a class of antibody. Okay. Okay. Anyways, IgE binds to the surface of mast cells like a little receptor almost. It goes out and interacts with all these antigens. And then it goes and releases granules and everywhere that it's near. So its little granules contain things like histamine. So that's the idea of how this is going to be working. Now, as far as why you are reacting to these specific antigens, it has to do with the fact that your body has made antibodies, or essentially immune globulin receptors, B cell receptors, that match whatever that is, and it's not getting cleared out and being told that this is non-dangerous or something like that. Your body made your 8 billion different combinations, and that was one of them that got through. Okay, let's watch a video. You know, it's a lot of clicking involved in this. Yeah, go.
A type 1 hypersensitivity, also known as an allergic reaction, is an immediate and exaggerated immune response to a harmless antigen. Some people develop a reaction when exposed to allergens such as dust, pollens, animal dander, or penicillin. This hypersensitivity is mediated by IgE. Sensitization occurs when the antigen first makes contact with some part of the body. The antigen is taken up, processed by antigen-presenting cells, and presented on a class 2 MHC to T helper cells. The T helper cells produce cytokines, which stimulate B cells to proliferate and differentiate into plasma cells. In those with allergies, these plasma cells switch to secrete IgE antibodies specific for the antigen instead of the usual IgM antibodies. As IgE is produced, the IgE molecules attach via their constant regions to receptors on nearby mast cells. Mast cells contain granules packed with chemicals that can induce a hypersensitivity response if released. Once attached, the IgE molecules persist for many weeks. The individual is now sensitized to the antigen. When exposed to same antigen for a second time, the antigen binds to the IgE antibodies on the mast cells. To trigger a response, the antigen must bind to 2 adjacent IgE molecules, resulting in cross-linking. Within seconds, the mast cell releases histamine and other mediators of the inflammatory response from its granules, triggering a variety of symptoms such as increased mucus production, contraction of smooth muscle, and leaky dilated blood vessels.
Okay. So what isotype of antibody would be most directly involved in type 1 hypersensitivity? this allergy stuff is a type 1. So that's going to be IgE, which I hope that you guys were just paying attention to that. Okay, chemokines and cytokines. These are things that are going to send signals between the cells to tell them what to do. The principal chemical mediators produced by mast cells and basophils in these sensitivity reactions: histamine, serotonin, leukotriene, platelet activating factor, prostaglandins, and bradykinin. We saw in that other table previously that histamine, serotonin, and bradykinin were on that list. So there it is again. Whenever you are having an allergic reaction, here's a thing that happens. I mean, here you go. Like you essentially, blood vessels will dilate, your bronchioles can constrict, you can get a headache, you can get increased blood flow, causing skin manifestations like patchy red patches or even hives, you know, all of this stuff. And then that can cause you to need to use an inhaler, even if it's not anaphylaxis, even if it's not technically asthma. If you are reacting to it enough, that would be considered, you know, your bronchial tubes like constricting on each other. So you can't breathe. Okay, so hay fever, that's allergic rhinitis. I don't want to have to tell you guys again what rhinitis is. I ask you about a question on the exam, which of the following is the type of illness that, you know, is that Ebola belongs to, and you have choices between like hemorrhagic fever and allergic rhinitis. You know, here you go. So rhinitis is your nose and being inflamed. It's being affected by this. Okay. So seasonal stuff, any of the inhalants and stuff like that. Then we have asthma, which is characterized by periods of impaired breathing and bronchoconstriction. And it's usually, they can react very responsively and very severely to very, very small amounts of inhalant allergens, foods, and other infectious agents that can cause these as well. So atopic diseases can include, we do have some persistent diseases that can be categorized with this as well, not just allergy and asthma. We've also got eczema on the skin, which is due to atopic dermatitis, and it's skin inflammation, in case you don't know what dermatitis means. This occurs through ingestion, inhalation, and occasionally skin contact. Essentially, hearing is bothering me. Essentially what we're having here is a reaction on your skin to something that you've either ingested or come into contact with externally. And inflammation that appears through your skin. That's what eczema is. People are prone to that. A food allergy, you have an intestinal reaction to something. We might have vomiting, diarrhea, abdominal pain. We don't always have association with the skin and the respiratory tract, but those are pretty common. We are used to seeing those as far as shellfish and peanut allergies and similar things go. Drug allergies can cause stuff like this as well. You can have a mild rash. You can also develop anaphylaxis. Penicillin, sulfas, aspirin, and opiates can cause things like this that can be actually fairly severe. Then we have systemic anaphylaxis. Leading into that, it's going to be your, like I said, systemic. That's going to be your entire body. It's going to be affected by whatever it is that you were exposed to. We'll have a sudden respiratory and circulatory disruption that can be fatal in minutes. The allergen that causes it and the route that it got in can also be very variable. This is often, we see this with bee venom, and we can have this from injections of antibiotics or serum as well, if that's something that happened. And the sensitivity can last for decades after exposure. You have one reaction to it like that, and then for decades you will react like that every time you are exposed to the thing. So we can diagnose allergies through a few different kinds of tests, typically on the skin, and not all of them have to be, what I'm trying to say, on your skin. That's what like through injected, like they do little bubble injections into your skin sometimes, but doesn't have to be that. So we have the radioallergosorbent RAST tests. These are pretty common ones. They measure levels of IgE in response to specific allergens, and measuring, welts on the skin and all of that. So, we have a lot of stuff available for testing on those, a wide variety of very specific things to see what you might be allergic to. The leukocyte histamine release test, the LHRT, is something that measures the amount of histamine released from basophils in response to specific things. And that's usually done in vitro and not necessarily so they can do that with like samples of your blood. They don't have to do it with injections into your skin. So antihistamines, just not as many choices available, I don't think. Antihistamines are used to, tamper down the histamine response so that we're not having quite so much of that allergic reaction. We getting rid of all of those symptoms that we might see with rashes developing, as well as, itchy eyes, watery eyes, runny nose, and all of that stuff. So, but it can cause drowsiness. They do. I guess they do have some antihistamines that don't have the drowsiness effect because they don't cross the blood-brain barrier like Benadryl does. So Benadryl is diphenhydramine. You have an allergic reaction and you need to save your life. The best way to do that is like we say with EpiPen. So that's going to be an epinephrine or adrenaline pen. You inject it or inhale it. You can use them in inhalers as well. It reverses constriction of the airways, slows release of allergic mediators like histamine and stuff like that, and all that. So it can buy you the time to get to a hospital for continuing treatment, but it is not something that you're meant to do in place of treatment. So it happens to you, you need to use your EpiPen. You may need to use more than one dose. Get yourself to a hospital because it could still continue on. So you want to get to the hospital immediately. Don't avoid it. Don't think I feel better and go home. No. Okay, vaccines for allergies. We're using essentially the vaccine mindset to desensitize and hyposensitize someone to a specific allergen. This is how it's going to work. It's essentially down at the bottom, we're going to have the allergen be introduced. We have the B or the plasma cell that has IgG blocking antibodies that can block the allergen and bind up with the allergen so that the mast cell that has the IgE that was reacting to the allergen, it can't react with the allergen anymore. It's being blocked up by the IgG. So we want to produce these. That's the idea. Cool. Just trying to make sure I said everything. Okay, treatment and prevention of allergies. Obviously, you want to avoid your allergen, but if we can treat or prevent it, and eventually, hopefully, I don't want to say cure you of your allergen problem, but eliminate it, hopefully. Okay, steroids can prevent the synthesis of IgE by the plasma cell, so you don't make the IgE in response to the allergen. We also have monoclonal antibodies that can inactivate the IgE itself after the IgE itself has been formed. We also have drugs that can act on the surface of mast cells to prevent them from degranulating and releasing all those signal molecules. And then we have antihistamines, aspirin, all those things that can affect cytokines on target cells to dampen the response. And then, of course, we have IgG production to interact with the antigen instead of IgE. So that would be the cure or whatever. So these are treatments for allergies there. So we're going to move on to type 2 cytotoxic reactions. These are based on antibodies. And the big one that we're going to be talking about in this section is, blood group antigen reactions. Okay, so ABO antigen markers on your blood cells or your red blood cells. They are genetically determined. They're composed of certain kinds of glycoproteins. And you have 2 of 3 alternative alleles. You can have an A allele, you can have a B allele, and you can have an O allele. And then based on like how I had showed you with the Mendelian square, which I'm sure you guys don't know what I'm talking about, with like the mom and the dad, for example, I don't know what my mom's blood, or, I don't know much about my mom's blood type. I know she's A something. Anyways, so go ahead and say my parents are, my dad is a B. So we'll say he's a B and an O. And let's say my mom is an A with an O. So he got a B from, a mom, and he got an O from a dad. And my mom got an A from a mom, and a mom got an O from a dad. It doesn't matter really. Okay. These are our possibilities of what we could have as babies. We could have someone with an AB. We'll just bring them in like that. We have someone with a BO. We have someone with an AO. And we can have someone with an OO. This is all 4 blood types in one setting. So you could potentially have all 4 blood types, right? I say all 4, but yeah, it's all 4. Yeah, all 4 blood types right there in one sitting. You have your AB, you have your A. So if it's AO, AO is recessive. So it doesn't get like, it's not going to be, I don't know how to describe it. It's not, I would say not get expressed, but the phenotype of what you would see would be expressed as, A. So AO is A and BO is B. And we just put this as O. So AB is its own thing. So these are the blood types you can have: AB, A, B, and O. Okay. So these, whatever. So there you go. So those are how that would work out. So you can have genes, or you can have AA, and that would just be A, depending on what your parents were and what have you. So you can have 4 different blood types. There are essentially 3 different surface markers: the A, the B, and the O. The O isn't a thing that we are concerned about with regards to rejection and all of that. Okay. The A's and the B's are. So, yeah, so here's kind of what they look like and how they would be different. So if you had type A, that has this crap on it. And if you had type B, it has this on it. If you had type AB, it has both of them on their surface. And then type O only has like the shell marker or whatever, common portion. Right. So if you are an A, naturally, you carry anti-B antibodies. Just normal. If you are a B blood type, naturally you carry anti-A. If you are an AB, you don't carry any. And if you are an O, you would have anti-A and anti-B. Both. So you carry those antibodies with you at all times. That's like part of like your genetic makeup of what's going on with this whole system. Okay. So if I give you a transfusion with, and you're an A, and I give you a transfusion with B blood, your body is going to have antibodies against the B, right? And it will attack it. And that causes a whole slew of problems. So you get the idea. So if you're AB and I give you an O, what would happen? Nothing. If you're an A and I give you an O, what would happen? Nothing. You don't have anti-O antibodies. But, if you're a B and I give you O, nothing. So O is the universal donor type. AB, since it doesn't have antibodies against anything naturally, you can give it anything. So AB is a universal recipient. So yeah, so that's how that works. You use agglutination essentially to figure out what your blood type is. I mean, I'm not going to go too much into what this means, but essentially because we did not cause agglutination here, we did not cause agglutination there. So now we know that this person is O because they have anti-A and they have anti-B in their blood. So if they didn't, it would have clotted down there. So that's the idea with what is going on there. Yeah. So yeah. And then this person would be O positive because they have a positive, they don't have an antibody against it. So that's that. So that's the RH factor. The RH factor is where you're O positive and O negative. or whatever. So where you're positive or negative blood type. So this person is A negative because they had an antibody that allowed for this to stay non-clotty. So you know that they are anti-RH. So they must be RH negative. So they are A negative. Okay. That's the idea there, why this person would be anti-A. Doesn't have anti-A, they're A blood type. It was allowed to clot, and the antibody for anti-B allowed it to stay in suspension or whatever. So that's the idea of how that works as far as when they're typing you for transfusions. So what is going to go on whenever you have, a mix-up here? A type A donor and a type B recipient. They have anti-A antibodies. So we transfuse the B person with the A blood. Their antibodies are going to interact with the markers on the surface of those A blood cells, right? Then what happens? We know what happens with antibody, right? All those things, those 6 different things that it can trigger, it's going to trigger those 6 different things. And one of the things that's going to be going on is complement cascade. So we're going to be poking holes in all of these cells that you just put in here and releasing all their contents. It's going to eventually cause shock. We're releasing hemoglobin into the bloodstream, hemolysis, and all that. So it's deadly, can be. So which of these blood types is a universal blood donor? It can give packed blood cells to any blood type. The cells is the O. Don't care about the O surface, right? That can go to anybody. Hemolytic disease of the newborn. This has to do with RH compatibility. We have an RH negative mother, and the baby is RH positive. She has a first baby. She has a newborn. That's because the blood of that baby, the RH positive blood of that baby, wasn't interacting with the mama blood until later in the pregnancy. So it takes a while for you to develop antibodies against it, but she did. So she developed antibodies against the RH factor because she's RH negative. So she has anti-RH. RH negative. So she has anti-RH antibodies. The baby has RH. It's RH positive. So this RH positive baby is born and it's healthy. It's fine. There's no problem here. Okay. Maybe she doesn't know. Maybe nobody ever found out. I don't know what's going on with her life. This happens again. She has a second pregnancy. During the second pregnancy with the second RH positive child, remember how our antibody responses are a lot faster the second time? Once that baby starts developing, essentially, once that baby starts developing blood, the mom's body is going to start attacking it. And it's usually pretty late on in the pregnancy. Anyways, essentially, the mom's body, the anti-RH antibodies from the mommy are going to attack the RH positive fetus again. This is going to lead to a hemolytic newborn. This means that literally the mom's antibodies were attacking all of the red cells in the baby that she was carrying. This is often deadly to the fetus. So what we can do is treat the mothers with something called RhoGAM to prevent this from happening. Essentially, we're going to be giving her some anti-RH antibodies. Anti-RH. It's hard to read because I said she has anti-RH antibodies. That's what I'm trying to explain to you. But we're trying to give her antibodies against this, against these antibodies to block the antibodies. That's what I'm trying to say. I'm trying to figure out a better word. But yeah, RhoGAM, you get antibodies to produce against the anti-RH antibodies that you have. Just like we were doing with the allergens before is what I'm trying to say. It's pretty similar to that anyways. Not exactly the same. It's not exactly the same, but it's a similar concept. Type 3, moving on, we have immune complexes, essentially immune complexes. These are antibody in the system that will combine with excess antigen hanging out. They will combine together. A lot of times the body will eliminate those, phagocytes will come along and eat them up and break them up, but some of them get stuck and launched in the basement membrane of the epithelium, which is at the bottom layers of the epithelium due to, capillaries, being tiny and all that. So they'll get kind of stuck up in there. They get stuck up in tight places like joints and stuff like that. And then our body will come and react because it's seeing antibody hanging out there, that this is antibody and we need to respond accordingly. So then we get inflammation in those areas. Blood vessels, the lungs and the heart, the joints, the skin, and the kidney are the main areas that are going to be affected by those. We'll watch a little video about it. This is starting to sound familiar with like lupus, because that's exactly what I'm talking about. Lupus is a combination of things, but that is definitely part of what's going on with it. Let's put our captions in play.
Immune complexes consist of antigens and antibodies bound together. Although large immune complexes are engulfed by phagocytes and removed, intermediate-sized immune complexes are not readily removed. These intermediate complexes react with and activate complement. For example, complement activation in blood vessels causes basophils to degranulate, which causes vasodilation. Immune complexes entrapped in blood vessels or other tissues, such as kidney glomeruli, activate complement, which attracts neutrophils and causes them to degranulate. Neutrophils release enzymes responsible for tissue damage.
Okay, so what kinds of immune complex diseases might we see? One of the examples is serum sickness and the Arthus reaction. We do set these aside from anaphylaxis separately because they depend on IgG, IgM, or IgA, whereas like anaphylaxis depends on IgE, just, so you know. Otherwise, they are very similar. They require a larger dose of antigen than anaphylaxis would. And then their symptoms are delayed, often hours to days after the exposure to, whatever they're talking about. But the Arthus reaction is localized dermal injury, whereas the serum sickness is systemic. Okay, so that is something you want to be aware of. If I were to ask you which one was localized, that is the Arthus reaction, and the serum sickness is, it smells like barbecue. It's weird. I don't know. Probably out in the office. Anyways. So type 4 hypersensitivities. Type 4 hypersensitivities involve T cells. These are cell-mediated hypersensitivities. The type 4 hypersensitivities result whenever T cells respond to antigens displayed on self-tissues or transplanted cells. So this is also known as delayed hypersensitivity because it takes a while for your body to really get going with whatever's happening with this. So to develop the T cell response and to react to whatever it is. I don't know what it is. It's got to be out there. It's got to be. It smells like a fire. It smells like a. Sorry. Anyways, we have something called an infectious allergy. So whenever you are sensitized to like a pathogen or something, and then you can have this type of a response to it, it's considered an allergy to that thing, but you're sensitized to it and it's due to an infection. So that's what we see whenever we do the skin tests with tuberculosis, and they inject that tuberculin underneath the skin. If your body reacts to the tuberculin, you'll see the inflammation as a result of it. And then, that just means you were sensitive to it and you may have tuberculosis or be carrying it. So we can also do similar tests like this with leprosy, syphilis, histoplasmosis, toxoplasmosis, and candidiasis. I don't know why I'm asking myself why I smell fire whenever there's like the fire training place right next door. I don't know why that's probably where it's coming from. Contact dermatitis is an example of this. We're talking about things like poison ivy here. Contact dermatitis, we get these haptens from household and personal articles as well. So we have the resins from poison ivy and poison oak. We have the haptens from household articles. We're talking about metals. If you're allergic to like nickel or whatever metal, stuff like that. Detergents, perfumes, we have a delayed reaction typically to these things. You don't react to them like super close to your exposure to it. That is because it's mediated by type 4 hypersensitivity. Yeah, and you can have, this is typically a reaction that you can have as well to aspirin, penicillin, trimethaprims. So yeah. So this is just explaining like what happens in contact dermatitis. We get exposed to, let's say, poison ivy. We have lipid soluble chemicals absorbed by the skin. Poison ivy oils are absorbed into the skin. Then the dendritic cells, which are, specialized monocytes that have adapted to live in like the skin, that's dendritic cells. They're phagocytes. They come into the epithelium, they pick up the allergen, they process it. T cells that have been previously sensitized to the poison ivy thing recognize that processed and presented allergen that the dendritic cell is showing it. Then the T helper cell will activate and secrete certain cytokines. The cytokines will attract macrophages as well as activate cytotoxic T cells to the site. And they'll come in and elicit a response that's exactly similar to what they would do if they were reacting to a pathogen. They just don't know that they're doing it for a stupid reason.
Harmful effects produced by the mechanisms of cell-mediated immunity are referred to as delayed hypersensitivity. Delayed hypersensitivity is mediated by Th1 cells, also called TD cells, and is responsible for contact dermatitis, tissue damage in a variety of infectious diseases, and rejection of tissue grafts. A contact hypersensitivity, such as poison oak dermatitis, is initiated by contact with the antigen, which is also called the allergen. During contact, a small molecule from the plant, called a hapten, binds to a carrier protein in the host. The carrier protein with bound hapten is ingested by a macrophage, and the hapten peptide is then presented on the surface of the macrophage on a class II MHC. Th1 cells with T cell receptors capable of recognizing the hapten peptide antigen interact with the presented antigen, become activated, and increase in numbers. When the host comes in contact with the allergen a second time, the Th1 cells react with the hapten peptide being presented on the macrophage and release cytokines, resulting in attraction of more macrophages, followed by inflammation and skin lesions. Characteristic skin lesions appear after 24 hours, reaching their peak at 48 to 72 hours after the second exposure to the plant.
All right, so T cell mediated recognition of foreign MHC receptors. So this is where we're going to be getting into graft rejection. So there's two different ways that you can reject a graft. So on the left, we have host versus graft. Essentially, we have put a kidney into someone's body, right? So here's an example of a kidney cell that would be associated with that kidney. A T cell receptor from the host comes in and can recognize the kidney cell from the donated kidney via the MHC1, and then that will cause it to, activate that. What cytotoxic T cells do by now, right? They release their perforins and the granzymes and they kill. So that's what's going on. The T cells, the helper cells, and macrophages can be involved in helping stimulate that response as well. It takes a while to build this up. That's why we don't know necessarily right away that somebody could be rejecting a graft. It can take a while. However, the second case that we're dealing with here is graft versus host. In this case, we're talking about grafted bone marrow. If I were to transplant like a femur into like, or a part of a femur into your leg or whatever, and it comes along with bone marrow, or you get even a bone marrow transfusion, something like that, the lymphocytes from that bone marrow will come and attack your cells and start an attack on your own cells. So that's naughty too. There's 4 different kinds of grafts that you could potentially get if you're getting a transplant. The autograft, which is from yourself. The isograft, which is from an identical twin. The allograft, which is what we typically think of, might be 2 different people. Your mom to you or whatever, anybody that's not identical to you. And then a xenograft being a transplant from a different species, which some people forget that we do that. Pigs heart valves and bovine heart valves and stuff like that are pretty common example of that. So transplantation of every organ has occurred, including the brain at some point in history. The most common transplants include the skin, the liver, the heart, the kidney, the coronary artery, cornea, bone marrow. Bone marrow is a pretty common one. So, and then sources can come from live donors who are like actually, donating their organ. I come to you and I say, I have a kidney, I can help you out and stuff like that. Or you can also donate skin that way, bone marrow and liver, part of your liver. We know about donating bone marrow while you're alive, of course. Cadavers can also be sources of these transplant tissues. We can use cadavers for heart, kidney, and cornea. When we're saying about heart and kidney, as well as we can get them from the livers from this situation. We are in lungs, of course. We're talking about brain dead people, whereas like cornea can be harvested from an actual, declared dead individual. No more brain activity. So yeah. And then we also can have donations via fetal tissues. You can get stem cells from like the placenta and stuff too. So which of the following is not a result of antibodies? So we know IgE for type 1, that's a regular allergy, what we think of with an allergy. Type 2, that was with the blood group thing. We have antibodies like anti-A and whatever. That was definitely. Type 3, that's where the immune complexes. And then type 4 is the cell mediated, and we're talking about T cells mostly, right? So that would be the answer would be D. So autoimmune diseases, we've kind of stepped a little bit into the concept behind these. These are anywhere when an individual develops a hypersensitivity to him or herself, things, proteins, cells, whatever it is within yourself. Autoantibodies, T cells, and sometimes both can be able to mount an attack against you. So that can be systemic, involving all of your organs, or it can be organ specific, involving only one organ or tissue. And this can be recognized. We have this, there are more than 80 recognized autoimmune diseases that can affect 5 to 8% of the US population. So these are some of the diseases here in this table. If you guys want to look through this, I'm not going to go through the details necessarily of these, but this is interesting to know. And I would be aware, I would read through this table and be aware of how each one would work, because we did talk about systemic lupus, erythematosus. That is SLE, that's lupus. That was the type 3 where we had and that makes sense, right? This is definitely the case with like rheumatoid arthritis as well. We see some aspects of type 3. It seems like type 4 and type 2 can be associated with that as well as with ankylosing spondylitis. Graves' disease in the thyroid, myasthenia gravis in the muscles are immune complex. Type 4 diabetes is a cell-mediated attack on insulin producing cells, and then multiple sclerosis has to deal with type 2 sensitivity as well as a type 4 sensitivity. Yeah, so you're welcome to look through the characteristics involved in these, but I would definitely be aware of the concepts behind how these work, at least, and that we that this is like, I don't know if you knew, I'm assuming you knew that lupus was autoimmune disease, but you should probably be aware of that right now. So is type 1 diabetes. Okay, autoimmunity, forbidden clones. We have the clonal selection theory where that immune system of a fetus develops tolerance by eradicating all self-reactive lymphocytes, right? We talked about that in the beginning, should be getting rid of them. Some of these may survive, and B and T cells may inappropriately attack tissues with self-antigens as a result of that. So that's the idea of how that would work. We also have the concept of molecular mimicry. We have the immune system misidentifying antigens because like microbial antigens can display molecular determinants similar to normal human cells. So we attack the microbe, it looks like our cells though, so now we're attacking our cells as a result of it. So that happens, we see that with rheumatic fever. I'm sure that makes sense, right? I don't know if you guys know about rheumatic fever, but that follows, that tracks as far as what I know about rheumatic fever. Anyways. You can also have the infection, the reaction to the infection causing cross reaction to the tissues around where the bite's being happening. And then we have we'll get into a little bit of specifics here. We have type 1 diabetes, insulin secreted by beta cells of the pancreas, regulating glucose utilization are being attacked by cytotoxic T cells. We think that might be due to a reaction to mumps, maybe perhaps either mumps itself, or maybe you might not have known you had it, or vaccination with it potentially. It was unsure there. That's an unsure thing. Yeah, rheumatic and lupus. So which of these below is not an autoimmunity? We just talked about lupus, rheumatoid arthritis, and multiple sclerosis. So please tell me that you know that tetanus is not autoimmune. We know it's caused by a toxin produced by Clostridium bacterium. Okay, immunodeficiencies, where your immune system isn't working well enough. Primary ones are present from birth. They are genetic. Primary are genetic. Secondary are acquired afterwards, usually caused by natural or artificial agents. Okay. So this was talking about SCID, and SCID where that the bubble boy happened. Essentially, we are affecting the pre-T cell and pre-B cell development there. There are some kinds of immune deficiencies, like when we get out here to the ADA and to George and all of these that come on a little bit later, but I don't know much about these. Hypogammaglobulinemia, not making enough antibodies, but some of these can be managed pretty well, and others aren't really that well managed. So yeah, so if you have deficiencies up here on the top, you'll get I'm trying to write on the table. If you have deficiencies up here on the top, then we have likelihood in fungal, protozoan, and viral infections, whereas this one down here, we have more likely to have bacterial. And if you have kind of both going on, which is the case with SCID, then that is, and it's X-linked. It's due to a gene that's on the X chromosome. That's why we see it more commonly in men, because they only have one X chromosome. Anyways, cause bacterial versus the protozoan, fungal, viral stuff. They have both for SCID, since both of the precursors are affected. So SCID, severe combined immunodeficiency. Boy in the bubble, no lymphocytes at all, no B cells, no T cells, cannot have cytotoxic T cells, cannot have antibody at all. So extracellular pathogens that are introduced to the body will grow unchecked and it will cause you to die. You cannot recover from it. They will not ever be able to be eliminated by your body. You would have to be taking drugs or something to eliminate it. So that's how that works. There's this terrible movie with John Travolta, which is probably better than the Jake Gyllenhaal one, which was awful, but I've seen it. I hate to admit. I feel like I saw it like, I don't know, on HBO, like back when it was on HBO, like a long time ago, you know? Anyways, so which of these following are referring to the boy in the bubble disease? That's SCID, that is C. Okay, secondary immunodeficiencies in B and T cells are caused by either infection, non-infectious metabolic diseases, chemotherapy, or radiation. We often think about that, infection with HIV causing something like this. And then chemotherapy and radiation being kind of an obvious one as well. So AIDS is the most recognized infection-induced immunodeficiency disease that we have studied. T helper cells, monocytes, macrophages, and antigen-presenting cells will be infected with HIV, and then depletion of the T helper cells and the functional impairment of the immune response as a result account for cancer, opportunistic infections, and stuff like that, that can eventually, if left untreated, lead to death. Bone marrow and lymphoid organ cancers can also cause malfunction in both the humoral and the cellular immunity, because you might be overgrowing like your white cells, your T and B cells. But let me ask you something. Do you think that those cells are behaving properly? No. So you might have a lot of them, but they're not doing their job. So that can lead to, a lack of function of your immune system as a result. So leukemia can cause this. We have massive numbers of cancer cells that will also displace normal cells in the bone marrow and the blood. You have to overgrow with those leukemia cells, those white blood cells, and the other ones aren't going to be able to function properly. Plasma cell tumors for large amounts of non-functional antibodies that your body doesn't need, wasting energy, wasting resources, and then thymus tumors for severe T cell deficiencies. So that's it for chapter 16. We talked about hypersensitivities, type 1, 2, and 3 dealing with antibodies, and type 4 with cell-mediated. Type 1 was the allergies, type 2 was cytotoxic, antibody-associated stuff. We're dealing with blood groups, and rhesus factor is Rh factor, which is what we specifically talked about in that category. Type 3 due to immune complexes, and type 4 delayed hypersensitivities due to cell-mediated reactions, and how that relates to autoimmunity. And then we talked about hyposensitivities, like the primary and the secondary ones, the genetic ones and the acquired ones. So that's that for that chapter. And then we have chapter 17 left. And then after that, you've got your unit 3 exam, and then we can move on into unit 4. So I'm looking forward to it, and I'm sure you are too.